DNA sequencing is the process of determining the order of succession of the 4 nucleotides that make up a DNA fragment. Specific sequences of nucleotides correspond to specific genes, which are the units responsible for encoding an organism's characteristics and transmitting them to its offspring. Sequencing consequently makes it possible to map the genome, that is, the complete genetic heritage that characterizes an individual or species. The whole genome of many species of animals, bacteria and viruses is now available in public databases. The sequence of an organism's genome can, as a result, be compared with known genomes in order to identify the species to which the DNA in question belongs. For this reason, DNA sequencing is performed in healthcare settings. Its purpose is to identify pathogenic microorganisms and study their molecular composition. It therefore contributes to the diagnosis of infectious diseases and provides useful information for defining strategies to control them. It allows, for example, to reconstruct the dynamics underlying the spread of microorganisms and to identify mutations that can render antimicrobial treatments and vaccines ineffective. DNA sequencing is also applied to the field of food safety, particularly to identify food fraud, that is, to establish when a food which ought to correspond to a given species has been substituted by a similar species of lower value. Next-generation sequencing technologies have been evolving since the early 2000s. The term refers to the range of DNA sequencing technologies that share the ability to simultaneously sequence millions of DNA fragments. It permits large genomes to be analyzed at considerably lower costs and in a fraction of the time taken by conventional methods.